Allison Cole. Feel free to call me Allie if you see me around town. I am with CERC, and I work right next door at the Alutiiq Marine Institute. I manage our biology laboratory space in our Chugach Regional Ocean Monitoring Program. I'm going to talk mostly about HABs and HABs monitoring in the Chugach region, but I'll give a little bit of like full program overview just to give a little bit more context of why we do what we do over there. So Mike already went over this. The only thing I'll add to this slide is that you'll see all of our communities are coastal, and so marine resources are very, very important in our region. And, oh, now it's going to be so blurry. Oh, there it is. Okay, good. In the Western kind of world, when we think about subsistence, we often think about like hunting, gathering, and that is part of subsistence, but there isn't really a good translation for what that means in Sukst in the Sugpiaq language. And so this is the closest translation, and it is our way of living. And so it's not just like what we're eating, how we're getting it, it's how we're processing it, how we're interacting with the land, and it's a really deeply ingrained in our culture, our language, our stories, dance, everything to do with our people. And so when you look at this poster, this is like a traditional foods poster. It goes from spring all the way over to winter, and when you look at these different sections, you'll see what people in the region might typically be harvesting during that time of year. And what you'll notice is the whole year round, we can go to our waters for marine resources. So it's really, really important that we have access to those and we know what's going on in our coastal waters. And so with all that background and context, we can really only imagine how horrible and traumatic the Exxon Valdez oil spill was for our communities. This event decimated beaches, killed billions of animals, and our elders today were the people who are outside of communities cleaning up those beaches. And some elders will refer to it as the day the water died because of what they were witnessing out in the landscape those days. And many of you probably know, I don't know if you do, in our region, there's various beaches where you can dig today and still find lingering oil. And so from that event came, a concern with knowing what's going on in our waters right outside of our communities. So that's how we established the Chugach Regional Ocean Monitoring Program, or the CHROME program. It's funded through the Exxon Valdez Oil Spill Trustee Council, called EVOS from now on. And it's really meant to, fill gaps in understanding coastal health, particularly related to seawater carbonate chemistry, harmful algal blooms, and then biotoxins from those algal blooms that can accumulate in shellfish. So here's kind of a diagram and overview. Our monitoring program is made up of citizen science samplers. There's one sampler in each of our 7 communities. We have one here with us today, which I'm so happy you could make it. Our samplers go out weekly and collect a phytoplankton tow, a seawater sample, and then a blue mussel sample. And they'll also, during that time, collect environmental data. So we have like a bigger picture of what's going on during these sampling events. Things like air temperature, water temperature, was it raining, what was the tidal state? So we can get a nice clear picture of what was happening during those sampling events. And then along with our community samplers, kind of like our core program, we also like to use this program to really strengthen capacity for ocean monitoring both in the Chugach region and with partners outside of the region, and use this as like an opportunity to build meaningful partnerships and collaboration. So for example, we work with the Alaska Harmful Algal Bloom Network very closely. We work with Prince William Sound Stewardship Foundation. They'll collect opportunistic samples, blue mussel samples, when they're doing summer projects out in the Sound. We also work very closely with the Ketchikan Bay National Estuary Research Reserve in Ketchikan Bay. So for example, this summer there was a couple of harmful algal blooms that happened in Ketchikan Bay, and working really closely with them to make sure that communications are happening, sampling's being covered, samples are getting coordinated and processed in a timely fashion. And I have a photo here of Riley. She was in the room earlier. Hi, there's Riley. Riley is recently stepped into the role of environmental coordinator with CERC, and that's really important because her job is really helping maintain these communications both with our samplers and with our partners. So for example, Riley actually helped us set up an 8th sampling location within the region in Whittier. So we're now getting that kind of western side of Prince William Sound covered really well with samples. Sierra. I'm shouting out everyone here today. So I'm not going to go too far in the weeds with this. I know there was an ocean acidification question. I would highly suggest speaking to our chemistry lab manager, Sierra Lloyd, right over here. All of our chemistry seawater samples, like they said, go to Sierra for processing. And she's got a really good grasp on how those impact our subsistence resources in the Chugach region. Okay, now we'll get into it. HABs monitoring in the Chugach region. So just a very quick, high-level overview. When we think about harmful algal blooms or HABs, we can kind of categorize them into these two groups, which is non-toxic, and when these bloom or start multiplying, there's just so many of them that it starts choking out other species in the environment. And then there are algae that can produce toxins, and so we specifically monitor for Pseudonychia, which can produce domoic acid and cause amnesiac shellfish poisoning, and we monitor for Alexandrium, which can produce saxitoxin or paralytic shellfish toxins. It's a whole group of toxins that can cause paralytic shellfish poisoning. And these algae can kind of exist in this dormant state as cysts down in seawater sediments, and in the perfect conditions, let's say there's a spring storm, it kicks up these sediments, and now these cysts have access to sunlight and warm water and nutrients, and they can start multiplying, and from here they'll enter the food web. And a lot of times we see them entering the food web through filter feeders. So these are neurotoxins. Shellfish don't have a central nervous system, and so they can just really either eat it and spit it out, or eat it and hold on to those toxins, and they're not going to be impacted like marine mammals or seabirds or us humans who might go and eat these shellfish. So I have blue mussels here. We use those for our regular weekly monitoring because they're kind of known for toxin depuration, so they can kind of take those in and clear them out really quickly. We also will take in subsistence harvested shellfish though for regular monitoring if people want that tested. So there's some butter clams over there. Those are a good example. Butter clams are known to hold on to toxins much, much longer. Think like months, years plus. They can really hold on to these toxins. And so Yana and I, Yana, our lab tech, Yana and I received blue mussel samples and those phytoplankton toes into our lab space. I'm not sure if you're familiar, but our whole biology lab, all the HABs testing used to be done in a 40-foot connex. So now our new lab space is all built out over there. If you go to the facility, it looks like a garage, but in the back, it's like a fully equipped biology lab. It's really wonderful to have this nice space. So this first room, it's like the main room you walk into. It's where we do all of our dirty work. We're taking in shellfish samples. We're processing those samples. We're doing toxin extractions, microscopy, and then prepping reagents for various assays. So this is kind of our main prep room. And then in the back, there's two specialized rooms. I'm going to talk about this one for quite a few slides. So this room is fully dedicated to toxin testing. So we run an enzyme-linked immunosorbent assay. We call it an ELISA, and that looks for domoic acid. So that's the one that comes from Pseudonychia. So that's that top, those yellow wells there. That's an ELISA, and we're using that to look at domoic acid concentrations. And we also run a receptor binding assay for those paralytic shellfish toxins, and that can tell us the total concentration of those toxins. So it gives a much better, comprehensive result compared to the ELISA. So we can see a bigger picture when we use that RBA. And I always like to just make it very clear, CERC or API, Chrome, we don't regulate shellfish harvest. We don't want to go into communities and tell them you can't eat something, and it wouldn't fly if we tried to do that. So we just test, and we can inform tribes on what's going on and what we're seeing, the types of results we see. Each of these toxins that we monitor has a federal regulatory limit. So for saxitoxins, that's 80 micrograms per 100 grams. And they made it nice and fun and used a different unit for domoic acid. That's 20 ppm. These are the allowable amounts determined by the federal government to be in food. So this is really important because for commercial shellfish industry, they're required to test for that, and they can get shut down if they're finding toxins above those levels. And there isn't really the same requirement for subsistence use, and not the same requirement, and there's not really the capacity in the state to be doing that for all subsistence use areas. So we monitor that in our region, and if something comes up from our regular weekly samples, or if someone sends something in, we go directly back to the tribes. So we put together these reports, we reach out directly to tribal leadership and provide that information so that they can make informed subsistence harvests. We do on occasion too, like I said, when we were working with the Research Reserve in Ketchikan Bay, we'll help kind of like develop or share language that's more like general public. So we can help do that too. We'll get into some of our results here. So this is the first kind of half of our field season for this year for domoic acid. I haven't got to updating my graphs just quite yet. But as you can see, there's really low levels. Not sure, maybe it's 20 parts per million is the federal regulatory limit, and our highest one that we've seen this season is 0.35, and that's pretty consistent with what we see, year to year in this program. Pseudonychia is really common in coastal areas all over the place. Domoic acid is really common in low levels in coastal areas, especially in Alaska, in these low levels. And so that's kind of started to kick off that conversation of, is there risk for chronic low-level exposure? So even though this toxin isn't above this limit, if people are eating it all the time and there's constant low levels of toxin, is there a risk from that? So there's been a lot of good conversations starting up recently about that. Here is a glimpse at our past saxitoxin data for the region. We don't see it commonly over that regulatory limit, but when we do see it, for example, there's one sample here that was over that limit. The following year we saw it in the same place, same time, over that limit. So it typically, when it almost colonizes certain areas, and so when those temperatures are right, when they get the right nutrients, they'll kind of start blooming same time of year, same place. And then it's the Seward Science Symposium, so I'm going to point out the Seward graph. It's a little small, so we have April, May, June, July, August 2023 are these ones here. And you can see a little bump here. And then not as significant, but this is April, May 2024. It is present in our samples, not as much. And again, I haven't updated this, but I have the data from this year, and there is another little peak again right during that same time of year. So we'll put a pin in that, and we'll kind of keep returning to this saxitoxin curve happening in Seward. Here's what some of our microscopy reports look like. So this is from those water samples, the phytoplankton toads that come in. We want to turn them into something digestible, so when we put out reports, people can look and say, okay, Seward, here's the samples that came from April, and here's the harmful algae that was in them. And these ones are specific to Alexandrium on the top, and then Pseudonychia on the bottom. We also keep our eyes out for dinophyces. We don't do any toxin testing, but dinophyces can produce okadaic acid. It commonly gets misconstrued with food poisoning. So we don't have dedicated funding to monitor for that yet, but we want to keep track of this so that if that comes up, if tribes want us to start monitoring that, we have this kind of background data that helps justify getting funding to monitor for that. And I'll just shout out again Seward up here. So remember we saw, or I guess we didn't see it here, but I let you know. There is, from this year, saxitoxin. It's not over the federal regulatory limit, but we didn't see any of that Alexandrium in the water. I'm going to carry this theme forward kind of through the end. We'll just glimpse at this really quick. We kind of take presence, absence of other common species to South Central Alaska. We've been taking this data for quite a few years now, and so trying to just turn it into something digestible, because it's just really a lot of information. So this is my first kind of go at trying to turn that into something you can glance at and see a good picture of what's going on outside of our communities. Okay, and this is that second dedicated laboratory space that I mentioned. Dustin Carl, he's not in the room, he's on a phone call next door. He's our Tribal Fish and Wildlife Director, and he established this environmental DNA lab. We do qPCR in here, and he established this with funds from the Administration of Native Americans to look at a traditionally used subsistence species And so he was using environmental DNA, taking water filters throughout the spring to look to see if he could see herring coming in and out. So we did it in Seward, and he collected samples this last spring from Cordova, so we're working on getting all this validated, so you'll hopefully see some data coming out about that in the next few months to a year. It's a slow moving, this one's a little complicated. I wanted to throw this in here. If those tanks look familiar to any of you, the Sea Life Center very graciously let us come in and sample from their tank for this herring project. They have herring in this tank and they let us come collect water samples as a positive control. And then they also donated fin clips for a really positive control. So this is Dustin collecting at the Sea Life Center tanks. If there's any Sea Life Center molecular scientists out there and are interested in collaborating, reach out to either me or Dustin. We'd love to have a discussion about that and do something like this again. I think could be really fun. And these are kind of the next steps in HABs monitoring. So like I said, we were seeing these kind of peaks of saxitoxin in Resurrection Bay, but not really seeing the algae ever, and we're not totally sure why. We don't know why that could be. And so having a tool like eDNA and our molecular lab space is really, really important so that we can really confidently and accurately say we're seeing this toxin at the same time that we're seeing peaks in this phytoplankton. So I think that's a really good example of how we could use a tool like environmental DNA. And then just a little bit of housekeeping to wrap up. So the last couple of years we've been producing this data from this program. Pretty preliminary data. Everything gets reported to the EVOS Trustee Council and is made public. You can access those on the AUS website or through the APMI website. And then we're working right now currently to get those wrapped up in a really digestible report and send those out with data directly to tribes so that they don't have to be clicking through links and navigating tabs. Those will just get sent directly to them by the end of this month, and we'll start doing that very regularly. And then moving forward over the next few years, doing a more comprehensive analysis, so using some of that environmental data and HAB presence to potentially start building out models and looking at HAB forecasting. And yeah, overall, the program is meant to be building this baseline data to inform our tribes of what's going on in waters directly outside of our communities and perform regular monitoring to inform food safety, food security within the Chugach region. Koyana. Is there any question? Oh, yeah.
How do samples get transported to you guys usually?
That's a really good question, because whenever we give this, I'm always I feel like it doesn't come across right, if it's too in-depth to go into in the talk. So typically what we do is we'll send out 8 weeks worth of sampling supplies in an action packer. So it's kind of one of these black totes. We package them up really well so that the glass doesn't break and sear. It happens sometimes. Sometimes you lose a sample. And so they'll take 8 weeks of samples at a time. And so it seems like this nice thing, we get weekly samples and we test them, but typically it's throughout the year, every couple months, we'll get a box of weekly samples from 2 or 3 months of sampling. And we just ship them. And then some of our communities don't have USPS, and so then it takes a little bit more coordinating of going to the airlines that do fly to the communities. That's another important thing to know. A lot of our communities don't have roads, it's boat or plane, so then it's coordinating with the pilots and the people who can transport things back and forth. Yeah.
Are there any ideas for why there's this disconnect between the toxin detection and the algae, or lack of algae detection? Just that people are thinking of?
I don't know that I've heard anyone mentioning anything. That doesn't mean there isn't. Yeah, I don't know. But I'm that's the best. I'm I don't want to just start making things up on the spot. We'll have a brainstorming session here. Yeah.
Do you ever process samples outside of the Chugach region?
Yeah. So, let's see. Oh, I think I forgot. There was a sentence in there that I forgot to mention. So, We do, like, our tribes, right, for this like core program, we also work really closely with tribes outside of the region who are really interested. Most of them send their HAB samples to Connect tribe, actually. They process through the ADEC, and then they work really closely with Sierra to get their water chemistry samples processed through her to kind of get that whole like piece of the puzzle going. So we do that for tribes outside of our region, and then we do also participate in, like, Mahmoud had mentioned it, some of our kind of research capabilities have been building up the last couple of years, and Chromis helped support some of those. Like, for example, we were helping collect samples for the eDNA. We're looking at different larval stages, and we're getting ready. We're waiting for Rachel Eichnoff. She's coming down to like process those, so we'll process those ones in-house. We're processing some of the samples from UAA from that like hydrocarbon study, looking at cortisol. I've taken some other samples in from Dr. Doug Hazy at UAA to look at domoic acid and saxitoxin. So yeah, we're all over the place. Oh, yeah.
I have a question.
Yeah.
If I were to go out clam digging, just for, get my own clams to eat, could I send them into you to get tested before I, serve them to my family?
Yeah, great question. And yes, you can. I'm like trying to think of how to, because yeah, we will always, so like we have this regular monitoring, and like I said, like we'll get like weeks worth of samples at a time. If we get subsistence harvested shellfish where people want to know what's in them, those get top priority. They get pushed to the top of the list over our like baseline monitoring program. Like obviously those are more important, and we want to run them really quickly because those conditions can kind of change. So we want to try and get anything that's a subsistence harvested run first and foremost. Thanks, Ian. Well, if there's no more questions, thank you so much.